Okay, hello IB Biology. This is Mr. McGee, and I'm going to try to teach you today about the electron transport chain that we've been talking about for a while. And this is one of the most complicated subjects with all of cell respiration, so it is very paramount that you make sure you are taking a look at your outline and you are reviewing this topic. First of all, this is the outline. I hope you've all had a chance to kind of look it over and glance. Before we get into the electron transport chain, just a review of how we got to this point. So if I go back to here, recall that we have the mitochondria with the Krebs cycle inside, as well as the electron transport chain. The glycolysis happens outside of the mitochondria in the cytoplasm. And of course, we do get 2 ATPs from glycolysis. So I'm going to type this in here. We get 2 ATPs. I'm just going to blow this up so it's a little more sizable. And if you recall, glycolysis is going to split into 2 pyruvates. That's going to lead into the Krebs cycle, which of course is going to cause the Krebs cycle to go through 2 separate turns. Each turn gives us 1 ATP. That means we get a total of 2 ATPs. By the way, you do remember that glycolysis technically gives us 4, but because we sacrifice 2 of them, we get a net product of 2, but that's why you may see it written as 4 or 2 depending on such. Well, at this point we've made some ATP, but we have not made all the ATP and even close to what we will get with the rest of cell respiration. But it isn't just ATP we've gotten up to this point. We have created electron-carrying NADH, if you recall. And so this is NADH created from glycolysis. It's going to go all the way to the mitochondria and get ready for the electron transport chain. In fact, you may recall that the Krebs cycle also gives us this, which then goes to the electron transport chain as well. But the Krebs cycle also gives FADH2, which for all practical purposes is going to have a very similar role to this. So we have all these electron carriers. They've been sending this whole time up until the electron transport chain, preparing, if you will, for final battle. That leaves us with the ATP that we're going to create here. And it is not 2 as the rest of them, but rather 32 ATPs that we are going to create. So we're going to take a look at the electron transport chain. But unlike all the others, this is the most important because it is producing the vast majority of the ATP that you and I use to keep alive. So let's go ahead and take a look at this remarkable process. But again, please make sure you understand up until this point what is happening here. Now, just a quick reminder, the mitochondria has 2 membranes, an outer membrane, which again is just like your cell membrane, the phospholipid bilayer. And then, of course, you have an inner membrane which is folded up like this. And its folds suggest more surface area. That is exactly what is taking place here. The inside of the inner membrane is called the matrix, and it is a more gelatin type substance. And this is where the Krebs cycle and the mitochondrial DNA are found. The electron transport chain is taking place, as we mentioned, along this inner membrane, and with all these folds, it's going to give us a lot more surface area. So, again, for a test question, why do we have the cristae, all of this folds? And the answer is more surface area for more of the electron transport chain. And then we're going to take a look at this gap here, this space, if you will, between the outer and the inner membrane. This is called the intermembrane space. And even though it's relatively insignificant outside of this, it plays a major role. And that is, we're going to pump hydrogen ions from the matrix all the way into the intermembrane space. So I'm going to go ahead and animate this here and show you what has taken place. I'll try to blow the screen up here so it's a little bit more evident, but make sure you're watching closely so you can see what's happening. Before we get to that point again, just a reminder, in glycolysis we created the 2 NADHs. I forgot to mention 2 were created in the link reaction, and then of course you get 6 NADHs and 2 FADH2s created in the Krebs cycle. Okay? And again, on my notes, make sure you take a look and see the basic layout, and that's what we're going to look at here. So this is the electron transport chain built along the inner membrane of the mitochondrial cristae. This is the intermembrane space, And then we have the mitochondrial matrix down here, and this is, of course, where the Krebs cycle all takes place. Now, the Krebs cycle is going to produce, as we saw, 6 NADHs, or it's going to create 3 in each of its cycles. And it's going to create 2 FADH2s. And this isn't all, because you remember glycolysis gives us 2 of these as well, and the link reaction gives us 2 of these as well. So at this point, we should have a total of 10 NADHs and a total of 2 FADH2s if you did all the math. But I'm just going to use these ones here to kind of signify this. And I'll move these oxygens and water off because we're not going to use that just yet. You'll notice we have these hydrogen ions, which are floating down here. And we talked about these briefly last year, but just a reminder again that hydrogen being the first element on the periodic table, it does not have a neutron. It's made of just a proton, which I'll illustrate this here. Hydrogen just has a single proton, and it has a single electron. And this electron orbits around the hydrogen atom like this. So that has the 1 positive and negative charge. Well, in the case of a hydrogen ion, if you think about it, we actually get rid of this electron like this. And I'm going to sketch that in there. And what we're left with out the electron is just the positive charged proton of the hydrogen. That is just one single proton. And that's why in chemistry we can refer to a hydrogen ion as a proton. That's really all it is. And if you scroll to the bottom of my notes here, I try to illustrate this concept. For those of you that get it, you can move on, but I put this in here because, again, I know not all of you have a strong chemistry background. It's basically just a single proton, but we call it a hydrogen ion. These are down in the matrix. Now let's go ahead and start this chain. We have these enzyme complexes here. And if you can see, 3 complexes have pumps in them. Complex 1, 3, and 4. Complex 2 is not a pump, but it's still going to accept electrons. So you can probably take a moment and make a prediction. What do you think is going to be pumped through these 3 complexes? And the answer is these little guys are going to get pumped through there. So you can also make another prediction. How are we going to get energy to make or to pump those hydrogen ions through there? And the answer, of course, is NADH that we have created. We are going to use these electrons from here, and we're going to use those to pump the hydrogen ions. Okay? Now again, complex 1 and 2, 3, and 4 are all enzymes, but we're just going to call them enzyme complexes, just to kind of make it short. And I probably could say well, I guess I'll say this right now, but one reason that the electrons are going to go on there has to do with the principle of electronegativity. So I'll actually, well, I guess I can illustrate this right now. I'll do it really quick and then we'll go back and review this here. The concept of electronegativity is simply the definition that an atom has the ability to grab an electron. If you think about it, some atoms really prefer an electron. They are popular with electrons. And some atoms are less popular. They don't really want the electrons. The more electronegative you are, the more you have popularity with those electrons. And so we're not going to get into all these different cytochromes here, but this chart shows some have a higher degree of electronegativity. But I'm going to make this even more simple for those of you that have a basic understanding of chemistry. Electronegativity is the ability to attract electrons. Rule of thumb, hopefully you recall from your chemistry class, SOL question here. The farther right you go on the periodic table, the more electronegative you get. The farther up you go on the periodic table, the more electronegative you get. And we, of course, have to exclude the noble gases. They don't count. And so if you look here, the most electronegative element is fluorine. But we also have oxygen and we have chlorine. Now this is going to come into importance later, but why are we using oxygen? And could we use something else as a substitute? And yes, fluorine can do what oxygen can do, and it can do it even better. But I'll explain later, fluorine is actually rare on this planet, and that's kind of why our bodies did not evolve to use anything but oxygen. But just be aware, these are some electronegative elements, and they are going to attract oxygen. However, complex 1 is also electronegative, and that means it's going to grab onto an electron. So let's go ahead and start the electron transport chain. I know that's what we're all here for. Here's NADH. It's floating around in the matrix. What it's going to do is it's going to go up to complex 1. And of course, remember, this is all Brownian motion. These are vibrating around. There's no guide in here. They're just bouncing around at random, and your body temperature provides this. But it's going to come up to complex 1, and because complex 1 is more electronegative, just a little, it's going to suck the electron off of the NADH. And again, it's not a big affinity, but because it's kind of like if you're fighting over something, the stronger person's generally going to win. And that's what complex 1 is. It's stronger. And it's going to grab onto that electron, and poor NADH now converts back to NAD+. And of course it disappears. It's NAD+. I just didn't animate that. That electron is now going to transfer energy, and we're going to pump a hydrogen ion through complex 1. And that's it. Now, if you recall, I said this last class, we have a molecule here called coenzyme Q, very similar to coenzyme A and acetyl coenzyme A. Coenzyme Q is kind of like a little fairy. I'm oversimplifying this, but that's kind of what it is. It's a little fairy complex floating through here, la-di-la-di-la, and it is more electronegative than complex 1. So when it happens to pass by, it doesn't really have any reason to, but that electron is suddenly more attracted to coenzyme Q, and it jumps aboard. And of course, coenzyme Q doesn't really care. It's just like, oh, well, I'm popular. Who cares? Do-do-do-do-do. And coenzyme Q kind of moves on in between these complexes here. That's really what's happening. But on its way over to complex 3, we have something else happening. Complex 2 has FADH2 move up to it. And again, make sure you understand that. NADH goes to complex 1, whereas FADH2 goes to complex 2. Technically, if you really want to get into it, FADH2 is actually made in the Krebs cycle within complex 2. FADH2 doesn't really float around down here. It actually is made within the complex. But for all practical purposes, don't worry about that. The electron will be released to complex 2 because it is more electronegative. And now here comes coenzyme Q. It's floating on by, and again, because it has a higher electronegativity than complex 2, this electron is going to pop on there and hop aboard. And now coenzyme Q has two little friends, whee, as it goes back and forth like this. And you guessed it, once it gets up to complex 3, it is more electronegative than coenzyme Q and more electronegative than complex 2 and 1. This is going to make these two things have, well, they're going to leap right to here because, again, of that higher affinity. It's a popularity contest if you think about it. I mean, if you go to a concert and then somebody cooler comes along, you're going to jump, ship and go to that concert. And then if somebody more cool comes along, you're going to go to that concert. And that's just kind of, well, I mean, that's kind of the best analogy I can make here. But now that these electrons have jumped ship to complex 3, we added energy to that. We are going to pump more hydrogen ions through this tunnel. And you got to remember, we're actually doing this several times each time here. And there's, if you remember, 10 NADHs doing this, so I'm oversimplifying this here. Now, once we've pumped those hydrogen ions to the other side, now we have those electrons sitting there, but here comes another fairy floating around called cytochrome C. Cytochrome C has an even higher electronegativity, kind of attraction. It's even more popular for electrons, and oh my goodness, these electrons are excited and they jump aboard the more popular and cool cytochrome C. And of course, cytochrome C floating around is really cool because he has those electrons, but then comes up to complex 4. By far the most cool and popular of all, and when I say that, I mean it is the highest in electronegativity. It is the strongest attractor to electrons, and they're going to jump from there to there. So again, up until this point, Make sure you understand why the electrons are moving from one complex to another, and it's because as we go along, one thing gets more electronegative than another. That's pretty much it. So if you make a plan for a certain date on a calendar, and then something better comes along, you're probably going to cancel the plans and go to the better event. And then if something better comes along, you're going to cancel those plans and go to that event. Well, that's the same thing happening here with these electrons. They're just jumping ship to something better that has more electronegativity to it. Now that we're at complex 4, we're going to pump some more hydrogens across to the other side. Okay? And by the way, we're just going to keep doing this for all of these here. They're going to be pumping through, and of course they only pump to the other side when there's electrons moving in there, but I just kind of wanted to make that clear. When the electrons bond to them, they transfer that energy, and that allows the hydrogens to pump through. At the end of the electron transport chain, we see what we've done is we have piled up all these hydrogens on one side, and we've created a gradient, kind of like osmosis, if you will, or kind of like diffusion. In fact, that's exactly what's going to happen, and we're going to see that in a little bit here. So let me move these off to the side. So before we finish this page up here, make sure you understand what you're looking at right now. We have the electrons on complex 4. But there's nowhere for them to go. And as they pile up on 4, we're going to get more NADHs that are going to come along, and they're going to add their electrons, which then going to, of course, jump ship to the next one and pump more. And we're just going to keep piling electrons on complex 4. And now you can see what the problem is. Complex 4 is very electronegative, but after a while, complex 4 just can't handle it. It's kind of like if you go to a concert or wherever you go, and if it's popular, eventually it's going to get crowded, and now it's going to cause problems. The problem is, after a while, too many electrons are going to neutralize the charge, and complex 4 will not be any more electronegative. And any additional electrons coming along, they're going to have nowhere to go, and thus the entire chain will stop. So, complex 4, it is the most electronegative, but as electrons pile onto it, eventually they will have nowhere else to go. And that brings us to one thing more that is more electronegative than complex 4. And if we look at our chart here, we can see complex 4, which is actually listed right there. And there's one thing that is far more electronegative than complex 4. It is actually oxygen. Far more electronegative. In fact, it is one of the most electronegative things out there, aside from, of course, fluorine. But, of course, fluorine is not that common on Earth, and we didn't evolve to use it. Here comes oxygen. It is the coolest of them all, extremely popular with these electrons. And here comes oxygen gas into your cell, and it was created from plants. And when this cool, hip oxygen comes along, suddenly all of these electrons jump ship. In fact, oxygen is so electronegative, it can take a load of these just piling onto it. Oxygen's got no problem. It's so electronegative, it can handle a huge amount of electrons. It's very popular, and it has a large capacity to do this. And here goes oxygen. Right once it grabs onto that, it's good to go, and it can float away. So that is the purpose of oxygen. Please make sure you understand that. Does oxygen add energy to our cells? Because I hear this claim all the time that oxygen powers your cells. But does it really? Oxygen, all it's doing is sucking away the electrons. That is all it did. But I don't want to deceive you here, because technically this is not quite the analogy. Once the electrons bond to it, it will chemically react with hydrogens, and actually it converts to water, H2O. This is where water is created within your cells, and now the water will float away because it's combining with the hydrogens that are left here and the oxygens. Okay, so let's go ahead and review this before we get to the last part. What is the purpose of the electron transport chain? We still haven't made ATP yet. What is the purpose? The overall purpose is to create a proton gradient in the intermembrane space. That is the purpose. Make sure you know that. The NADH is used on complex 1, whereas FADH2 is used on complex number 2. Make sure you understand that. And of all 4 of these complexes, which one is the most and least electronegative? Complex 1 is the least, whereas complex 4 is the most electronegative. Okay? And next question. Let me see here. Oh, what is the role of cytochrome C and coenzyme Q? You could say they act as ferries going back and forth, shuttling those electrons along, this chain, if you will. And the other question, what is the function of oxygen? And the answer is it is extremely electronegative, one of the most electronegative things out there, and as a result, it has a huge capacity to grab onto electrons and strip them off of that complex 4. When it does that, it converts into water, H2O, from those hydrogens that are down here as well. That is where we get water from. In fact, you can see these hydrogens here will bond and become the water. Okay? So the role of oxygen is to create water. And if you can see from here, there's a lot of misconception that the carbon dioxide you exhale from your breath was actually oxygen, but no, the oxygen you exhale actually comes out as water vapor when you exhale. We'll see later, but CO2 actually comes from decarboxylation of sugars and fats. That's where the CO2 comes out of your breath. Okay, so again, make sure you understand the role of oxygen and how it converts to water, and all the purpose here, which is to create all of these protons. And then we still haven't answered that question of how the heck are we going to make ATP, and actually that is the very last part here. So I know this is definitely a lot of stuff to cover. Let's go ahead and get through this here. This is the electron transport chain, which I'll divide it right down the middle here. We just talked about all this, but there's another enzyme complex off to the side here. It's called ATP synthase. In fact, you can probably guess from the name synthase, it's an enzyme that synthesizes ATP. But it also is kind of cool because it spins around at the bottom. At least when hydrogens go through it, it spins around, kind of like a water wheel. You know how they have on rivers to generate power? When a hydrogen shoots through here, it's going to spin. So let's see what this is. This is called chemiosmosis. Once we create this proton gradient, there's a lot of pressure and they can't go back down this way. But they can find an opening somewhere, and here it is, an ATP synthase. With all this pressure, they zip through the tunnel, wee, like this. And when they zip through the side, it causes ATP synthase to spin around, and that causes the phosphorylation of an ATP molecule. Here comes a hydrogen, and we just made an ATP. Whoops, just dropped my microphone. And here comes another hydrogen, wee, floating through the channel. And what do you know? We just phosphorylated another ATP. In fact, we will keep on doing this. We get a total large number of ATPs. And if you remember the total count, I'll just put it here as a reminder, we get a total of 32 ATPs generated. Okay? From that spinning ATP synthase. So please make sure you know chemiosmosis. It's kind of part of the electron transport chain, but it's not really. Chemiosmosis is where we take all those protons, we run them through the enzyme, it spins it around, and that provides the energy for phosphorylation. Okay? And that leads me to the last part, and this part is probably one that's hard for people to grasp when they don't really have a full grasp on it. But it is, where does the energy come from? Well, if you remember in glycolysis, the enzymes make the ATP, but it's just the enzymes simply do it from the heat of the environment. Because it's just an enzyme being used like normal, we call it substrate level phosphorylation to make the ATP. Again, it's because of the energy of the environment. Same thing with the Krebs cycle. We have enzymes, and they just use the normal energy from the environment. It's called substrate level phosphorylation. So you could argue the heat from your body heat is enough to provide activation energy for your body's enzymes. But the next one here the electron transport chain. This enzyme, and again, I know this is confusing, but think about it. This enzyme is not working because of your body temperature creating Brownian motion that, makes chemicals react. This enzyme is working because hydrogens are shooting through it, and that causes it to spin. And if you think about it, the hydrogens got here because they were put here by the oxidative power. All that oxidation of those sugars put those electrons here, and that caused the hydrogens to go through. So the energy for this enzyme actually came from the oxidation of those carbon molecules from sugar. And therefore, this phosphorylation is far more efficient. It's called oxidative phosphorylation because of the oxidative power of an electron. On the other hand, when we look at, again, glycolysis in the Krebs, the enzymes just simply are coupling with substrates and it creates the ATP. Not a big deal. But this enzyme, again, it's not really coupling with anything. Well, it is coupling, but the energy is coming from the hydrogens, which is coming from the electrons in your body. Therefore, we call this phosphorylation oxidative phosphorylation. Again, lectures can only explain so much here. Make sure you take a moment to read about electronegativity, which I put here at the bottom, and also this process of chemiosmosis and what is taking place here, okay? And likewise, what's the difference between oxidative and substrate level phosphorylation? On the last slide here of my outline, I explain the difference between NADH, FADH2, and all of those that we are creating. We get a total here of 36 ATP that are generated in this entire process. So with that said, please make sure you are focusing on everything, and well, that's really what it is. And you can see here I've got the vocab list, so make sure you are taking the time and focusing on this here. All right, any questions, you can please let me know at any time of your convenience, but otherwise I'll let you guys take a look. One other thing I'd recommend you do is just sketch this out somehow. Make some cutouts or something of squares, see if you can lay it out on a piece of paper in your house, make yourself an ATP synthase, and really follow through what we just did here. Okay? I will review this later. In fact, your Monday asynchronous assignment, I'm going to give you this review chart here so you can practice with it. Okay? Otherwise, hope you guys have a fantastic day, and I will see you all later. Good luck, and thank you very much for your patience.